Hey everyone, JJ here. Welcome to another biochemistry lesson. In today's lesson, I'm going to be talking to you guys about the lysosome and mTOR signaling, and more specifically, mTOR signaling and the role of the lysosome in mTOR signaling regulation. So just to kind of give you guys a brief background, so the lysosome was first discovered in 1955 by Christian de Duve. It was originally thought to be the cell's garbage can, so it was just thought to be somewhere where the cell would just kind of get rid of its waste, degrade old damaged proteins. But more recent evidence suggests that in addition to these things that the lysosome was always thought to have done, it also seems to be important in sensing the nutrient status of the cell. So that brings me to mTOR. Now, mTOR is the mammalian or mechanistic target of rapamycin. So that's the fancy name for mTOR. It was just found that it was a target of a chemical known as rapamycin, and it comes in two complexes. One is called mTOR complex 1. And its major component that differs in this complex is the presence of a protein component known as raptor. And there's also mTOR complex 2, which has the protein complex known as Richter instead of raptor. So mTOR is the master controller of protein synthesis in the cell, and for protein synthesis to occur, you need energy and nutrients, and indeed mTOR is regulated by energy in the nutrient status of the cell. So even just looking at this now, you can see, okay, if the lysosome is an important nutrient sensor, well then there must be something, or somehow these lysosome and mTOR are connected in some way, and in fact they are. So just to kind of quickly go over the schematic of how this occurs, say we've got normal autophagy in the cell. In another video I'll be talking about autophagy, so check out autophagy in one of my other videos. So autophagy is just autophagy, which is just self-eating. That's kind of the actual name of this process, but it really just means that the cell is degrading proteins, its own intracellular proteins, and other intracellular organelles and other components within the lysosome. And it does that by collecting these components in vesicles that are known as autophagosomes. And what it does is it actually delivers the autophagosomes by a process known as macroautophagy to the lysosome, whereby the autophagosomes fuse with the lysosome, depositing the content, such as proteins, into the lysosome, where lysosomal proteolytic enzymes known as cathepsins actually degrade this protein into amino acids. Now here's where it gets interesting, guys. The amino acids are a nutrient, as we all know. So in fact, the intralysosomal nutrient or intralysosomal amino acid content will actually regulate mTOR, and this is how it's done. So amino acids in the lysosome actually activate a protein known as regulator, and regulator is composed of several different subunits, but we won't get into that in this video. But just know that amino acids regulate something known as regulator. And there's another protein as well I didn't mention, but it's also important in this process, and it's known as VATPase or vacuolar ATPase. So we'll talk about that in another video. But just for now, we'll look at regulator. So amino acids activate regulator, and regulator will actually activate RAG proteins. So there are actually 4 different types of RAG proteins: A, B, C, and D. And these isoforms actually form heterodimers with each other. So A and B will form a heterodimer with either C or D. So you could get a RAG A and C heterodimer, or a RAG A and D heterodimer, or a RAG B and C heterodimer, or a RAG B and D heterodimer. So there are 4 different possible heterodimers that RAG proteins can make. And these two proteins are actually a part of something very important in the lysosome, which is Linus machinery, or lysosome nutrient sensing machinery. So LY for lysosome, NU for nutrient, and then S for sensing. So Linus machinery, these two proteins are involved in Linus machinery. So that's just the way the lysosome actually senses and monitors interlysosomal nutrients. So what will happen, guys, is that when regulator gets activated, regulator is actually a GEF, and a GEF is just a guanine nucleotide exchange factor. And what that means is regulator will actually GTP load RAG heterodimers to activate them. Now I'll get into this process a bit more in another video. It's kind of a complicated process, but I'll talk to you guys about it in another video. But just realize that regulator is a GEF. It GTP loads RAG heterodimers and it activates them. And what it'll do is it'll actually enable the RAG heterodimers to actually bind to some intracellular proteins. And one of those proteins is in fact mTOR complex 1. And now mTOR complex 1 will in fact actually migrate to the lysosomal membrane and bind to RAG protein. And then once it does that, it becomes activated by a number of things, but I won't get into that so much in detail in this video. But just be aware that mTOR complex 1, it binds to RAG proteins. It gets activated. It gets activated by a couple proteins. One of them is Rheb protein, RHEB. I'll talk about that in another video, but it enables its activation when it's bound to RAG at the lysosomal membrane. Two other proteins that regulate mTOR complex 1 activity are AMPK, or AMP activated protein kinase, and AKT, or otherwise known as protein kinase B. Now, AMPK is a very important energy sensor of the cell. It'll actually be activated by AMP, which means that the cell is actually energy deficient. So when the cell is actually energy deficient, AMPK will be activated, and what will happen is it'll actually negatively regulate mTOR complex 1, or inhibit it, which makes sense, right? If the cell doesn't have a lot of energy, it's not going to want to put energy into the process of protein synthesis. It's gonna stop protein synthesis from occurring, because protein synthesis is an energy-intensive process. Now AKT is somewhat different. AKT is activated by other things such as insulin, and what AKT will actually do is it'll actually activate mTOR complex 1. So just realize that mTOR complex 1 is not only regulated by its binding to the lysosomal membrane, but it's also regulated by other factors such as AMPK and AKT. But nevertheless, if mTOR complex 1 is activated, what it'll do is it'll start to activate some of its downstream targets, such as P70S6 kinase or P70S6K. So it'll phosphorylate P70S6K. P70S6K will then actually phosphorylate the S6 subunit on a ribosome, activating the ribosome. And we all know that a ribosome is where proteins are actually made, or polypeptide chains are actually made. Now, for protein synthesis to occur and to work properly, you need initiation and elongation of the polypeptide strand. Now, there are other proteins that are involved. There are many other proteins involved, but a couple important ones. One in particular for initiation is EIF4E, or eukaryotic initiation factor 4E. Now, EIF4E along with EIF4G will actually activate and promote polypeptide synthesis initiation. So just you want to know that these proteins are involved in polypeptide synthesis as well. Now, not only are these eukaryotic initiation factors required, but there's also eukaryotic elongation factors, and one of them is EEF2. So eukaryotic, the lowercase e is for eukaryotic, and then this capital E is for elongation factor 2. Now that, as his name suggests, promotes elongation of the polypeptide strand when it's being made. Now, what will happen is these proteins, the initiation factors and this elongation factor, are actually regulated. The EIF4E and its binding to and its binding partner EIF4D are actually regulated by 4EBP1 or EIF4E binding protein 1. So they're actually negatively regulated. And what will happen is the 4EBP1 actually binds to EIF4E, which inhibits EIF4E's ability to bind to EIF4G. So it's kind of complicated, but just know that 4-ABP1 actually inhibits EIF4E. Now, EEF2 is also regulated by an upstream kinase called EEF2K or EEF2 kinase, which actually inhibits it. So EEF2K will actually phosphorylate and inhibit EEF2. Now, in order for mTOR to complete its process and promote protein synthesis, it has to deal with these negative regulators. And what it does is, because mTOR complex 1 upregulates and activates P76K, P76K is actually a negative regulator of EEF2K. So if you negatively regulate, if you inhibit an inhibitor, then you're going to activate the protein involved. So P76K actually inhibits EEF2K, so it actually inhibits EEF2K's ability to inhibit EEF2, which means that EEF2 becomes activated. I know it's complicated, but that's how it works, guys. And then the other negative regulator that mTOR has to deal with is 4-ABP1, and what it actually does is it actually directly phosphorylates 4-ABP1 to inhibit 4-ABP1 from inhibiting EIF4E. So again, because mTOR C1 inhibits 4-ABP1, it actually will end up activating EIF4E and EIF4G, which will enable initiation of polypeptide synthesis at the ribosome, all of which will end up leading to polypeptide synthesis. So that's how the whole process works, guys. Anyways, guys, that was a brief lesson on mTOR signaling and the role of the lysosome in mTOR regulation. If you found this video helpful, please like this video. It's greatly appreciated, and please subscribe for more lessons. It's all greatly appreciated. All your comments, all of your feedback is greatly appreciated. Thank you so much, guys, and have a great day.